So hello dear all students and welcome to another lecture from Genome Biotech. In this video tutorial, we would be talking about how the cancer progression and development takes place in accordance to molecular biology view that how the genes are regulating the progress in the development of cancer that is taking place. So as I have already told you, the cancer was referred to as an uncontrolled proliferation of the cells that were dividing. Okay, so the a cell that has escaped the normal body metabolic functions of apoptosis, cell cycle regulation, or differentiation leads to the development of a cancer cell. Now, in this lecture, what we have to see is that explain the difference between the benign and malignant tumor. I will be explaining you what are the benign and malignant tumor, describes how the tumor progression takes place, in what manner the tumor are going to be progressed, summarize the properties of the cancer cells, and then compare cancer induction by chemical and viruses, that how the chemical induction of cancer takes place by different chemicals as well as viruses that will be inducing the formation of cancer. Okay, so types of cancer. Firstly, we are going to classify the different types of cancer. So the cancer can be divided into several different types. One is referred to as a tumor, a benign tumor and a malignant tumor. So basically we are dividing the tumors into two different types, the benign tumor and the malignant tumor. Okay, so I'm just going to categorize the tumor here into two different types. The tumors are being classified into two different types. One is referred to as the benign tumor, and another one is referred to as the malignant tumor. Okay, benign and malignant tumor. So one would be referred to as benign and another would be referred to as malignant. As explained to you, these benign tumors are the one that remains localized at a particular location within the body. They will remain in a particular location. These tumors are always capsulated. That means there is a proper capsule shaped structure present over these tumors. And that is why these are referred to as capsulated tumors. These are called as capsulated tumor because they have a capsule present over their structure. Okay, so there is a very well defined capsule that is surrounding these types of tumors, and because of this particular capsule that is surrounding these types of tumors, they are referred to as capsulated tumors and remains localized within this particular region. They can grow in their size, but then also they will always remain capsulated. These are always capsulated. Whereas as told to you about the malignant tumors, malignant tumors, sorry, about the malignant tumors are mobile tumors that may move from one location of the body to another along with the bloodstream. Supposingly, this is the bloodstream, then the malignant tumors have the ability to move from one location to the other. These are the malignant tumors which would be growing. And as these malignant tumors are growing, so they have the ability to form a new blood vesicles towards the mass of cancer cells, and then they would be moving. When they have the ability to move to new locations in the body, then this phenomena is referred to as malignancy. Malignant tumor always display the following two features. One is referred to as the angiogenesis. Angiogenesis. Angiogenesis refers to the formation of blood vesicles towards the mass of cancer cells. And another feature that these cells shows is commonly referred to as metastasis. Metastasis means the movement of cancer cells from one location to another location in the body is commonly referred to as metastasis. So these are the two features which are being shown by the malignant tumor cells. Okay. Now, moving forward. So, see here, these never remain encapsulated and these will always be. Now, cancer cells. Cancers can result from abnormal proliferation from of any of the different kinds of cells in the body. So any of the cells that are present in our body, they undergoes abnormal proliferation, then it will lead to the formation of cancer. A tumor is an abnormal proliferation of cells, which can be of two different types. It can be benign, it can be malignant in nature. Okay. Benign tumors, such as common skin warts. So any skin wart refers to any type of an lesson that could be present over the skin. They could be in the form of a vertical wart. Okay, skin wart will remain confined to its original location, neither invading surrounding tissues nor spreading to distinct body sites. So they won't be invading the surrounding tissues, nor they would be moving from one side in the body to another. Okay, so these are referred to as benign tumors. But when you talk about the malignant tumor, what do you mean by malignant tumor? Malignant tumors are the one that shows invasiveness. These tumors have the ability or capability to of both invasiveness surrounding the normal tissue and spreading throughout the body via the circulatory or the lymphatic system. So these are the tumors that have the ability to move from one location in the body to another location with the process of metastasis. So they can perform metastasis. They can move from one location to the other. Next, only malignant tumors are properly referred to as cancer. Whenever you talk about the malignant tumor, malignant tumors are commonly referred to as the cancer. These are the ones that are referred to as cancer and not the benign tumors. Now, see, tumors can again be classified into several different types on the basis of the type of tissues that are going to be influenced by the tumor cells. Just deleting all these things. Just a second. Clearing all the drawings and moving forward. Now, cancers could be divided into three different types. One is referred to as the carcinoma, which include approximately 90% of human cancers, are malignancies of the epithelial cells. These are the malignancy of the epithelial cells. Whenever the epithelial tissue is going to form cancer, then it is referred to as the carcinomas. Okay. Epithelial tissues refer to the skin tissues, etc. They are going to form the carcinoma. Then you have the sarcoma, which are rare in humans. And these sarcomas refer to the solid tumors of connective tissues such as muscle, bone, cartilage, fibrous tissues. These are all referred to as the sarcomas. Then the third one is referred to as leukemia and lymphoma, which account for approximately 8% of the human malignancies. Okay, this is the leukemia or the lymphoid tissue tumor is the one that is responsible for 8% of human malignancies, and this is the tumor of blood forming cells. So blood forming cells causes leukemia. The rarest of all cancer is sarcoma. Once in part B, a question has been asked about the rarest of all tumor, that is sarcoma. And carcinoma include 90% of human cancers as malignancies of the epithelial cells. Those are referred to as the carcinoma. Next, development of cancer. Now, how in what manner the cancer development is going to take place? See, cancer development, the development of the cancer cells takes place not in a single step, but in a series of reaction. The first step among which is referred to as tumor initiation. Tumor initiation is the first step of the process of cancer development. Is thought to be the result of genetic alteration. See, tumor initiation is being caused by the result of genetic alteration leading to abnormal proliferation of a single cell. Genetic alteration, as the thing indicate. So this is actually being caused by the initiation. Tumor initiation is being caused because of a particular mutation occurring in a particular cell. See, this is the cell that has undergone a particular mutation. This cell began to divide itself. As this cell has the ability, as the cell gains the ability to divide itself, so it has the ability to undergoes multiplication. Furthermore, mutations accumulate in one of the other cell among the population, and this cell which has further been mutated undergoes further division. This cell undergoes further division. As this cell undergoes further division, so it will divide, it will be dividing. As it will divide, it will lead to the formation of another type of cells. Okay, so this is the slow process. The tumors are being accumulated, the new tumors are being produced, making the most competent cells to outperform the rate of division of all other cells. Slowly and steadily, the accumulation of mutation takes place, and ultimately the cell began to move from one location to the other, leading to tumor progression and then tumor establishment. Now, how does tumor progression takes place? Cell proliferates, then lead to the outgrowth of a population of clone derived tumor cells. Tumor progression continues as additional mutation occurs. This is very important. Within the cells of the total tumor population, the cells of the tumor population that have been produced by the mutation within themselves undergoes further mutation. There is further mutations that are going to be added up, and there is tumor progression that is going to be continued as additional mutations occurs within cells of the tumor population. So addition mutations are being caused, and these addition mutations are being added up. As these additional mutations are going to be added up, so you will get new type of mutations being added, and the most actively dividing cell is going to then outperform the rate of division of all other cells, and it will keep on dividing, leading to the formation of cancer cells. Okay, then the cells displaying the cancerous property will be selected through clonal selection hypothesis. So now, after the division has actively taken place, then the second step that leads to the tumor establishment is clonal selection. Okay, so what happens in the case of clonal selection? Some of the mutations confer a selective advantage to the cells that are previously dividing, such that the cells began to move or divide much more rapidly, and the descendant of the cell bearing such a mutation will consequently become dominant within the tumor population. The phenomena is commonly referred to as clonal selection hypothesis. What happens in the case of clonal selection hypothesis is that that one of the cell which has been mutated, as the cell has been mutated, so what has happened? There is a mutation that has been formed in a cell. Now this cell undergoes further selection, further mutation, and a clone of cells is being produced. Then these cells undergo clonal selection, and one of the cell that has been undergoing clonal selection is going to rapidly divide, and then such type of cell will begin to migrate through the blood vesicles to a new site, leading to the formation of cancer. So the most active and the most efficient cell is being selected for the formation of cancer by the process of normal natural selection occurring in our body, and that is referred to as clonal selection. Okay, so you can see that if you talk about studies on colon carcinoma, if you are going to talk about the colon carcinoma, that is the colon of our intestine, okay, then you find here is that one of the cells within the proliferating population is then given thought to give rise to a small benign neoplasma or adenoma or polyp. Okay, so one of the cell that is dividing will give rise to a neoplasma or an adenoma or a polyp. So a polyp or an adenoma is going to be produced. Further round of clonal selection is going to take place, and further the division is going to occur. Next point. See, now we are going to talk about the properties of the cancer cells. First property of the cancer cell: the cancer cell do not display the following different types of properties. Now, which are the different properties? First, the normal cells will have cells which will grow in a particular density. Now, these are the normal cells which are present. These normal cells will undergo proliferation, and cell become quiescent when they come in contact with regard to each other. As the cell comes in contact with regard to each other, they will completely become quiescent. They will not further divide. Whereas when you look at the tumor cells, these tumor cells undergo proliferation. As these tumor cells began to undergo proliferation, they will lead to the formation of a layer of cells, and further on, each and every cell will divide over the other cell. These cells will begin to proliferate and will not have any type of a contact inhibition. There is no contact inhibition. Okay, so there is no density dependent inhibition. Normal cells proliferates in the culture until they reach a particular finite density, at which point, they become completely quiescent, no further division takes place. But cancerous cells or the tumor cells have the ability to undergo infinite rounds of division, so that these cells continuously divide. As these cells continuously divide, so they will lead to the formation of large number of overlapping cells. Okay. Next, properties of the cancer cells. I have already told you about the density dependent inhibition. They have no contact inhibition. Cancer cells never undergo any contact inhibition. Normal cells have contact inhibition. They comes in contact and they completely stops or halts their proliferation, whereas cancer cells undergoes proliferation. Okay, tumor progress as a result of mutation that leads to increased proliferation and malignancy. The progression of tumor takes place because of accumulation of mutations, and these mutations are being constantly added up. Thus the tumor began to progress. Okay, cancer cells continues proliferating under conditions that causes normal cells to cease and proliferate and enter into the G naught phase. Okay, so cancer cells are the one that undergoes proliferation even under such conditions that the normal cells will not be able to divide. Another property of cancer cells. Then loss of contact inhibition, already told to you in the diagram. There are the normal cells, they have contact inhibition, they cannot divide, but cancer cells are dividing one over the other. Another property of the cancer cell is that these shows autocrine growth stimulation. Autocrine growth stimulation means they can undergo their own stimulation to undergo division. They produces their own growth factor. Let us look at this diagram and let us explain this diagram. Now when you look at this particular diagram, then you say that the cancer cell exhibit autocrine growth stimulation. What is happening in the case of cancer cells? You look here. In the case of cancer cells, these cells are undergoing autocrine stimulation. Instead of receiving large amount of stimulus of growth factors, they are producing their own growth factors. You can look at this particular diagram. They are producing their own growth factors. As they are producing their own growth factors now, so that growth factor is going to spread and are going to interact over their own receptors. As these growth factors will be growing, they will be interacting over their receptors. Then what is going to happen? They will interact with their receptor and will cause the cell signaling to take place, so that the cancer cells will undergo division. Receptor stimulation takes place, then these cells divide. A cell produces a growth factor to which it and neighboring cells can respond. The cells will be producing growth factors. Growth factors will be responded by the neighboring cells, resulting in continuous secretion stimulation of the cell proliferation. It will result into continuous stimulation of several different types of cells, and all the different types of cells will be stimulated to express those particular growth factor molecules. This is how the growth factor molecules will be stimulating the cell to undergo further division. Next, so there is an autocrine stimulation. The cancer cells exhibit angiogenesis. Second, cancer cells secrete growth factors that promotes the formation of the blood vesicles. Now, which are the growth factors that are being secreted? As I have already told you that in the case of chapter number C, figure number 19.3. So we will be moving to that particular figure number 19.3. Just a second. We will be looking at that particular figure. Just a second. There's a very good figure 19.3 given that would be explaining to you. This is just a review. Okay. There was a figure in glycolysis about the hypoxia inducing factors. I'm not getting that figure. If I can get that particular figure, figure number 20.5. Okay, already shown to you. Figure number 19.3. But I'm not getting that particular figure. Okay, might be. So this the last topic that we were talking about was angiogenesis. Angiogenesis is needed to support the growth of a tumor cell beyond the size of a million cell, because it will have to form new different types of blood vesicles that are being required to supply oxygen as well as nutrition to the proliferating tumor cells, so that the proliferation of the cells can take place and these cells can undergo effective division, so that these cells will divide and will lead to the formation of effective tumor cells. The formation of such new blood vesicles is very important only in supporting tumor cells, but also in the case of metastasis. So tumor cells will undergo formulation of the blood vesicles, due to which they will be undergoing the process of metastasis to take place. Okay, so that they can move from one location to the other. Next. All the general characteristics of most cancer cell is that they fail to undergo differentiate normally. That means these will not be able to differentiate properly into different types of cells. They will not be able to differentiate themselves. Okay, so they will not undergo differentiation. The a programmed cell death or the process of apoptosis is also absent in the case of cancer cells. So, the process of cancer formulation and the properties of the cancer cells could include the following. You can just write their names, or as I'm telling you here, the properties of the cancer cells could include the following. First, cancer cells defies any density dependent inhibition. They do not have any density dependent inhibition. They do not have any contact inhibition. Loss of density dependent inhibition as given in Pathfinder, loss of contact inhibition as given in Pathfinder. Then these cancer cells undergo autocrine growth stimulation within themselves. There is the autocrine growth stimulation that takes place in the case of cancer cells. Then these cancer cells have the ability to undergo angiogenesis. That means they can lead to the formation of blood vesicles. And then the next point, these cancer cells undergo programmed cell death, which is also commonly referred to as apoptosis. These do not undergo apoptosis. These do not undergo terminal differentiation. Terminal differentiation is also completely absent in the case of cancer cells. Next point, causes of cancer. Now, what are the different causes of cancer? Now, basically, there are two basic causes of cancers that you can talk about. One is referred to as the chemical agents and the biological agents. Okay, so basically, the causes of cancer that I'm going to categorize here, I'm going to categorize the causes of cancer into three different types. The causes of cancer can be categorized into three different types. One are the physical causes, another one is referred to as the chemical causes, and the third one is referred to as the viral causes, the biological causes. Okay, so chemical, physical, and biological. So I'm just going to write them out in front of you. So these are the first and the foremost that we would be talking about are the chemical causes that can lead to the formation of cancer. Then we would be talking about the physical causes, and then the last one, we would be talking about the biological causes. Biological causes include the viruses which are being involved. Okay. Now, physical causes of the cancer will include the following two different types. One would be referred to as the ionizing radiation, and another would be referred to as the unionizing or non-ionizing radiation. Okay, non-ionizing radiations that would be present. One is ionizing, and another one is non-ionizing. Non-ionizing. Ionizing radiation include alpha radiation, beta radiations, the gamma radiations that are present, and non-ionizing radiation include the UV radiation, the X-rays which are present. Okay, chemical mutagens. All these cancer-causing agents are commonly referred to as mutagens. All carcinogens are mutagens, but all mutagens are not carcinogens. Okay, so mutagens is a very broad term. Sometimes the mutagen can be a mutagen, but it cannot be a carcinogen. but carcinogens are always mutagenic agents in nature. Okay, for example, UV induced mutations can be normally repaired in our body. They will not cause mutations normally in our body, but except in the case of a particular condition which is referred to as xerophthalmia, in which the UV radiation damaged TT dimers cannot be repaired because there is an inability to biosynthesize the repairing enzymes in our body. So there you will be getting the cancer, else not. Okay. Next point, causes of cancer. Substances that have the ability to cause cancers are referred to as carcinogenic substances. Okay, they have been experimentally identified, and epidemiological analysis has been done about these carcinogenic substances. These include several different types of chemical, biological, or the radiation. Many agents, like including radiations, chemical, and viruses, have been found that can induce mutation in both animals as well as humans. Now, what we are going to talk about firstly is about the carcinogenic agent, the chemical carcinogenic agents that we are going to talk about. See, carcinogenic chemicals include in tobacco smoke. Aflatoxins, a potent liver carcinogenic agent, is produced by some of the molds. Then there is the carcinogenic agent that is present in the form of tobacco or benzopyrene, dimethyl nitrosamine, nickel compound that are present. Smoking is the undisputed cause of nearly 90% of all lung cancer. More than 90% of all lung cancers are being caused due to smoking, and it's responsible for causing the cancer of the oral cavity, the pharynx, larynx, esophagus, and other sites. In total, it is estimated that smoking is responsible for one third of all cancers, death that are occurring globally. So it is responsible for more than one third of the cancers, death occurring all over the world. Clearing all the drawings and moving forward. Next. See, the first cancer which was being developed was by the Barry Marshall and Robin Warren when they firstly established a link between the Helicobacter pylori and stomach cancer. They induced themselves with Helicobacter pylori, the causative agent of the stomach ulcer. Marshall deliberately infected himself with the pure culture of the microbe and studied the course of the infection. For their great studies, they were being awarded Nobel Prize. They have been also being awarded the prize because they were the first pioneers to prove that there were biological agents that are responsible for the development of cancer type diseases. Next, about tumor promoters. There are other different types of chemicals. Those are referred to as tumor promoters. What do you mean by tumor promoters? Tumor promoters are those chemicals whose presence greatly or drastically increases the chances of occurrence of a tumor. Tumor promoters, since the increased cell division, they induce, facilitate the outgrowth of proliferated cell population during early stages of tumor development. So they will lead to the formation of tumor. They will be inducing tumor formation. Moreover, the cell proliferation induced by tumor promoters lead to mutation that unavoidable occurs during DNA replication. Carcinogenic agents act as a tumor promoters in human, including asbestos, some viruses that causes chronic inflammation, and the bacterium that is responsible for causing cancer is Helicobacter pylori. So all these are causing different types of cancers. Okay, several different types of viruses which are commonly referred to as tumor viruses are responsible for 10 to 20% of the worldwide cancer incidences. So these are all the different tumor viruses that are present. These viruses include both the DNA genome viruses as well as the RNA genome viruses. Some of the very important viruses from where the questions are always being asked is herpes virus causing Burkitt's lymphoma. Okay. Then you have the polyoma virus and the simian virus 40. which is responsible for Merkel cell carcinoma, hepatitis B virus, the liver cancer, the major cause of liver cancer, hepatitis C, which is again causing liver cancer, and retroviruses, which are such as HIV and HTLV. They are the one that would be causing the T cell leukemia. So you must remember all these five examples. Okay. So especially the hepatitis B, which is being a DNA virus, hepatitis C, which is an RNA virus. Now, so you must attempt all these questions. What are benign tumors? What are malignant tumors? What is their difference? Role of clonal selection in the development of cancer? How autocrine growth stimulation contribute to the formation of cancer? Cancer cells frequency? How they secrete proteases to digest the collagen? Okay. How does exposure to sunlight causes skin cancer because of the presence of UV radiation? And how can vaccine prevent liver and cervical cancers? Now, see, because the virus, when enters into your body, if antibodies are present in your body, then they will be attacking the virus and preventing its entry into the cell. So, the formation of antibodies can be induced by the presence of vaccines. So, vaccines can induce antibody formation in our body. Okay. Vaccines can induce antibody production, can induce antibody formation, can induce antibody formation in our body, formation in our body, as they can induce antibody formation in our body. So, will be helpful in combating these viruses, in combating these viruses, and killing them up. Okay, thus protecting us from infection. Next point, we would be talking about the oncogenes which are present in the case of cancer viruses. So, what are these oncogenes? How these oncogenes and the different types of viruses are having the different set of oncogenes that would be leading to the formation of the tumor in our body. So, we would be looking at these oncogenes in tomorrow lecture, because we will be finishing off these oncogenes tomorrow. And there are several different types of oncogenes, and the most probably this chapter would be over by tomorrow. I'm winding the class here only, but at least try to have a reading about the different types of oncogenes that are present in our human body, that can be proto-oncogenes that can be modified to form the oncogene. That's all for today. Hope you had enjoyed the lecture. Thank you. Good night.